organic compounds? Organic compounds are chemical compounds that generally contain carbon-hydrogen bonds. Organic compounds are usually held together by covalent bonds. Carbon has several properties that make it particularly useful to living organisms. For one thing, it can form bonds with one to thousands of other carbon atoms to produce large molecules that can have many different shapes. Due to this property of carbon, the body can build many organic compounds, each of which has a unique structure and function. Therefore, many organic molecules are relatively large and they have unique characteristics that allow them to carry out complex functions. The chain of carbon atoms in an organic molecule is called the carbon skeleton. The important categories of organic compounds include proteins, lipids, carbohydrates, nucleic acids, and adenosine triphosphate or ATP. Now, what are proteins? Proteins are large biomolecules that contain carbon, hydrogen, oxygen, and nitrogen. Some proteins also contain sulfur. A normal lean adult body is 12 to 18% protein. The monomers of proteins are called amino acids. So we can say that amino acids are the building blocks of proteins. There are 20 different amino acids in human body that can be combined to form proteins. Each of the 20 different amino acids has a hydrogen atom and 3 important functional groups attached to a central carbon atom. Proteins are complex molecules that are synthesized in stepwise fashion. One amino acid is joined to second amino acid and then a third amino acid is then added to these first 2 amino acids and so on. The covalent bond which is joining each pair of amino acids is called a peptide bond. When 2 amino acids combine, a dipeptide results. Adding another amino acid to a dipeptide produces a tripeptide. Further additions of amino acids results in the formation of a chain-like peptide containing 4 to 9 amino acids or a polypeptide which contains 10 to 2000 amino acids. So small proteins may consist of a single polypeptide chain with as few as 50 amino acids, and large proteins have hundreds or thousands of amino acids and they may consist of 2 or more polypeptide chains that are folded together. The sequence of amino acids in proteins is very important as it determines each protein's unique three-dimensional structure and its function. Proteins are present in all living organisms and they include many essential biological compounds such as enzymes, hormones, and antibodies. Foods that contribute to proteins are meat, fish, eggs, nuts, milk, cheese, beans, etc. Proteins can be structural and functional. In structural proteins, filaments form microtubules that provide cytoskeletons of cellular organelles. They form structural framework of various organs and connective tissues like hairs, nails, bones, skin, cartilage, etc. These structural proteins provide strength and mechanical support. They include keratin, collagen, and elastin. Functional proteins are mainly the enzymes of cell. They are biologically active substances that help support and maintain normal body functions. They carry out functions in the body unlike structural proteins which produces structures. Proteins perform many different roles in human body. They help repair and build body tissues. They allow metabolic reactions to take place. They provide structural framework to body and coordinate body functions. Proteins also maintain body pH and fluid balance. They are also responsible for transport of oxygen. Proteins also protect body against microorganisms in addition to providing your body with a structural framework. There are four levels of structural organization of proteins. First is primary level or primary structure. Second is secondary level or secondary structure. Third is tertiary level or tertiary structure. And in last, there is quaternary level or quaternary structure. Now, what is primary structure of proteins? The primary structure is the unique sequence of amino acids that are linked by covalent peptide bonds to form a polypeptide chain. A protein's primary structure is genetically determined, and any changes in a protein's amino acid sequence can have serious consequences for body cells. The secondary structure of a protein is the repeated twisting or folding of neighboring amino acids in the polypeptide chain. Two common secondary structures are alpha helices and beta sheets. The secondary structure of a protein is stabilized by hydrogen bonds which form at regular intervals along the polypeptide backbone. The tertiary structure of a protein refers to the three-dimensional shape of a polypeptide chain. Each protein has a unique tertiary structure that determines how it will function. The tertiary folding pattern may allow amino acids at opposite ends of the chain to be close neighbors. Several types of bonds can contribute to a protein's tertiary structure. The strongest bond is known as disulfide bridges. This bond is formed between the sulfhydryl groups of two monomers of the amino acid cysteine. There are many weak bonds that also help determine the folding patterns of tertiary structure, which include hydrogen bonds, ionic bonds, and hydrophobic interactions. Now, when we talk about quaternary structure, then in those proteins that contain more than one polypeptide chain, the arrangement of the individual polypeptide chains relative to one another is known as the quaternary structure. The bonds that hold polypeptide chains together in quaternary structure are similar to those that maintain the tertiary structure.